This is an overview of an article entitled HCN4 Gene Polymorphisms are associated with occurrence of Tachycardia-Induced Cardiomyopathy in patients with atrial fibrillation, published in the July 2018 issue of Circulation, Genomic and Precision Medicine. Tachycardia-Induced Cardiomyopathy, or TIC, is a reversible cardiomyopathy induced by supraventricular or ventricular tachyarrhythmia. Atrial fibrillation, or AF, is the most common cause of TIC in patients without a history of structural heart disease. Patient factors that increase vulnerability to TIC are poorly understood. In particular, no reliable genetic markers of TIC have been identified. Among the genetic loci that have been linked to AF in genome-wide association studies, or GWASs, of individuals of European descent, is a locus harboring the HCN4 gene. HCN4 encodes a hyperpolarization-activated cyclic nucleotide-gated channel that's highly expressed in the conduction system, where it's involved in heart rate control. In the paper under discussion, the authors hypothesized that HCN4 is involved in TIC pathogenesis and investigated the association between HCN4 single nucleotide polymorphisms, or SNPs, and TIC. The authors enrolled 953 patients with AF who underwent catheter ablation at Hiroshima University Hospital between November 2009 and August 2016. 73 of these patients were diagnosed with TIC, having initially had a left ventricular ejection fraction less than 40%, and then recovering the ejection fraction after AF ablation. 857 patients without TIC were used as the control group. The authors genotyped 17 SNPs in the HCN4 locus in DNA samples from the patients. They found that the strongest association was with the SNP RS71648883, which had previously been reported to be associated with AF in GWAS. This figure shows how all of the TIC patients began with an ejection fraction less than 40% and recovered to greater than 40% following AF ablation. RS71648883 was strongly associated with TIC, with a p-value of 2 × 10 to the -9th, and with an odds ratio of 3.3 for each copy of the minor allele of the SNP. Of note, the authors found that the SNP was not associated with AF in Japanese patients, in contrast to the original finding in GWASs with individuals of European descent. The figure here shows a variety of SNPs in linkage disequilibrium with RS71648883 in Japanese individuals, all clustered in the vicinity of the HCN4 gene. As shown here, the authors confirmed the association between RS71648883 and TIC in a replication cohort comprising 350 patients with AF who underwent catheter ablation at either of 2 Japanese hospitals between September 2016 and June 2017. In conclusion, the authors demonstrated that the minor allele of RS71648883 in the HCN4 locus was associated with increased risk of TIC in Japanese AF patients. This suggests a strategy in which AF patients found to have 1 or 2 RS71648883 minor alleles would be managed with early therapeutic intervention, such as stricter heart rate control or early rhythm control, in order to prevent heart failure. It should be emphasized that the mechanism by which the risk of TIC is increased remains to be determined, and the association warrants replication in independent cohorts, particularly in other ethnic groups.